Hey, the automatic unmute does work. So
Dang, I feel very significant whenever that little answers in atheism swipe was just like, am I significant or what?
yeah, it's pretty awesome. Had you seen that before, Andrew?
What?
Did you see the kid doesn't even know what's going on? This is gonna be great.
Hey, who's president? Who's president, Andrew?
Yeah.
Barack Obama.
Yeah.
Oh, we're in trouble. How many fingers am I holding up?
Hopefully.
16.
Oh.
Okay.
Bad.
So here we are. The thing I want to concentrate on is the Hangout Unimmutable did with Cell the other night.
Sure.
And that is what I'm interested in. And
I think or just to say something real quick, I think me and Sal actually agreed on something. If vaguely agreed on something. So that was cool.
Yeah, the phylogeny stuff.
Yeah, the nested hierarchy, that was a treat to hear. I kind of have more respect for him because of that. Not that I didn't already, but I have even more respect.
Yeah.
Well, be careful because I've noticed that Sal will, like in a subsequent video, will backtrack sometimes some of the agreeableness that there was. I think he did the same with Dr. Dan.
Yeah.
Well, just the fact that he said it alone is good enough for me.
Yeah, I guess I have an opinion about that, but I'm not going to share it.
Okay.
Just at this point. The thing I did notice about listening to Cell with S.J. Thompson and a couple other people is that you got to understand that no matter what the science says, Cell's about the Bible. He's not about the science. The science is secondary, and he will be that way for some time. So you want to keep that in mind, I think, when you talk to him, because I think it influences him a bit. Oh, let's see here.
This is always going to influence like scientists too, right?
Yeah, but okay, when you think about it, like how does
it's not as much, but
how does a natural worldview influence a scientist? So say I'm a scientist and I'm studying evolution of dogs, and I used this example the other day. So I'm studying dog breeds and how they broke off from wolves, right? And so I'm studying this and I'm trying to figure out the genetics of the evolution of dogs. And now we have a creationist who's also figuring out the evolution of dogs, right? How are me and him going to differ? I think the answer is not at all. We're going to use exactly the same scientific methods, the exact same theories, and all of that. But now suppose I told my creationist friend that now that we got dogs figured out, I want to figure out how they diverged from bear, from the bear clan, right? And raccoons. And then all of a sudden we're different. But now the question is, am I doing anything different in how I approach the subject or the phylogeny? And is the creationist going to do anything different? And I think the answer to that is that I'm going to do exactly the same kind of science. And my worldview is not affecting it at all.
Here's how I see the difference going. Is if you're just studying wolf to dog or something similar like that, then you're going to start out with a kind coming off that arc there, because that's already in their worldview. That already has to be a true thing for them.
Yeah, but
That's
so
not
does
a true thing for you.
yeah, but it's the same thing for me. I'm starting out with the wolf,
Right.
with the candidate. So I'm not doing anything different. We're both.
Well,
We're
are
both.
there any other
And
mammals
besides,
of from that family that aren't wolves or aren't what you would consider dogs?
hyenas. They're not sure about hyenas, are they?
Yeah. So
I'm not
there's
sure.
the thing. You have to start with the starting point has to be with whatever kind you can imagine that doesn't rule out any other mammals out there that are kind of cousin or sister or brother lineages.
Yeah, but
into
you got a little bit backwards, Scott. You
Oh,
don't start out with the common ancestor. You start out with the extant species. So we're starting out with what we have today,
okay, with wolves. All right.
and then we're going to go backwards. But the point is that I'm not doing anything different either, whether I'm studying the common ancestor of bears and dogs or wolves and dogs. I'm using exactly the same science. So
Right.
how is my worldview influencing
it's
me?
a matter of how far back you extrapolate, basically. How parsimonious the conclusion is.
Yeah, the creationists will get off the bus at the kind level, and other than that, we wouldn't talk about it at all. I could work with a creationist and not even know he was a creationist if we were just studying dogs and wolves, right? He would never do anything different in the science. So that is the response when they say that your worldview is influencing you. It's like, no, not so much our worldview. It's just that I don't have a stopping point when I'm going back in history and trying to put together common ancestry, right? Because they put together common ancestry too. They do it just as much as we do. In fact, they do it a lot, right? With dogs, the dog kind or the wolf kind. So I don't see their complaint as valid because of that. So I'm like, when I'm studying, like if I do study bear and dog, I'm not going to be thinking, oh, my worldview. I'm going to be thinking genes and proteins,
Well, right. Scientists don't deal with worldviews when they're
No.
doing science.
Yeah. So that's kind of the point. So anyway,
it's funny. Scientists are kind of like practical fact-making machines, but at the same time, they're also imperfect in terms of the conclusions they come to.
yeah.
So it's or I guess they're just human. That's the best way I can put it at this point.
Yeah, well, it's a mystery that you're trying to figure out,
Right. They're basically humans who want to be more objective.
Yeah.
I think that doesn't sound weird at all.
Yeah, we don't know everything, for God's sake.
Right.
Okay. So now, did you follow the stuff he brought up with immutable after he dropped off with? I think you guys kind of left it with phylogeny, right? And Cell kind of he admitted that the phylogeny was powerful evidence for evolution.
Right. Yeah.
Is that correct?
Ancestor.
Yeah. So, hang on a second. Let me make sure I got this muted. Okay.
I think we both agreed that calm descent and evolution, or the mechanisms of evolution, are kind of separate. So you can kind of they're related to each other, but they're not inclusive. Like they're kind of what one doesn't depend on the other.
Yeah. Okay, so I have a bunch of little clips, and I'm trying to. I want to kind of get rid of the chat. I see immutable was in the chat. So the first one is miracles, and if you got any other clips or spots that you want to stop at, but let me know if you want me to search for something. But let's go ahead and run this and see what he says here and see why I clipped it.
Because I don't think there was enough time. So
Sound good?
Yeah.
Sound check.
Yeah,
Okay.
sounds good.
Okay.
if one accepts the young Earth model, then there's just no time. The similarities then would be by design. If one accepts a old Earth model, one could say, okay, you know, I might like Dr. B. He accepts intelligent design, but he doesn't think that, and he accepts universal common ancestry, but he thinks there has to be intelligent design along the way. Now, he doesn't use the word miracle, but that's the way I would interpret it.
Right.
Is that there are these gaps. So so you may hear me say, okay, if we accept this model, this is not feasible. If we accept this model, this becomes feasible, but then there's still some issues there. So
Danakins. That's our new code word, Danakins. So he's saying that he's got Dr. Behe, who accepts old Earth creationism, and he accepts young Earth creationism. And he admits that young Earth creationists have a whole lot of work to do because they don't have any time. But even Behe would accept the same thing that a lot, I mean, he would go a lot back, he would go back much further on that timeline. And the person that doesn't have a religious view on creationism, you know, that believes it happened at the Big Bang, like a lot of Christians believe that, you know, that basically God created the universe in such a way that it had to create life, right? So they believe that. So it has nothing to do with your worldview. It just has to do with where you draw the line in the sand as to biblical interpretation of how old the earth is. Now, that has nothing to do with Christianity, because there are a lot of Christians, including, I think, the entire Catholic population, that accept an old earth, you know, an old universe, right? Am I correct?
I think so.
So there goes that complaint. So that crystallizes what Cell and SFT and Ramat the fruit bat have to do. They absolutely have to cram this entire model into 6,000 years. And that is a daunting task. And that's most of what they're doing is that. All these videos they have, it's all about cramming all of this stuff into 6,000 years and somehow making it work. So I just wanted to point that out. So,
Right. Well, I would,
you know, and I pity them. Don't you pity them?
Sometimes.
Yeah.
No, I don't really pity them because they're committed to just a ridiculous idea, and it's kind of harmful to people who should be turning to real science books rather than creationist books. So it's hard to pity them because I think they're doing damage.
Yeah, the damage.
40% of our population, at least.
Yeah, the damage is kind of unforgivable.
Well, you got to remember, Scott, that sometimes it's because of other people that have been before their lives, for instance, or not before their lives, like earlier in their lives, and that caused them to have that worldview.
Yeah,
that's true. They were indoctrinated early, at a time before their brain could be critical.
Yeah, but we all have friends that are like, well, or we know of people or heard of people that are like marathon runners, and they decide to devote their entire life to running like a fucking 100K marathon or something, and they have to work their asses off. So in that way, I pity them that they have a hell of a marathon to run here, to completely rewrite all of science and cram it into 6,000 years.
Right.
And, you know, but talk to me tonight on Saturday Night at the Atheist, and I'll tell you my other opinion about them.
Okay.
But I'm not doing that.
Sounds like a good reason to show up for SNATA if you.
Yeah, it's a good reason to show up for SNATA.
Yeah, if you're deciding on the fence anyway.
But you see what I mean? It's really, they have a daunting task. I mean, they have to work their asses off in order to make all this work, right? And Cell is he's one of the guys that's actually doing the real work on this job. So he's actually digging in and trying to make this work, right?
I think the difference is that they're going around saying that it fits together, and it's all neat and tidy, basically. And we think, of course, that's neat and tidy, but of course it doesn't all fit together.
Right.
Like it's there are some holes and some gaps in our understanding, and of course that's where new science comes in. So I think that's the difference is between admitting a theory's limitations when it does have those limitations.
Yeah. Okay, so my next bookmark is at 1419, and I have learning to know. I don't know why I called it that, but it's coming up in about 40 seconds, so I'll just play it.
Okay.
Yeah, we'll let it roll through.
So I'm expressing my personal views, and then I'm going to also say what I think is and is not consistent with a particular model.
Right.
And then, of course, the question is which model, particularly of the fossil record or the age of the Earth, is valid. So, and I'm happy to acknowledge, let's work within the models, because
Right.
I think there's we could still learn something and understand some aspects of things, even if we accept certain models just for the sake of argument that we may not personally accept. And
Right,
we
oh, sorry.
oh, go on.
Or my personal way of looking at it is that in order to learn, you have to know what people already know, if that makes sense. Like what the current standard is, I guess.
Well, even in mathematics, there's one of the most famous proofs was the square root of 2 is irrational.
Okay, I'm not sure how important that is, but he makes the point that they had to make an assumption in order to explain a mathematical model. You make certain assumptions, and then you find out if there's any contradictions.
Right.
And that's famously how I grown my naturalism. I assume complete strict naturalism, and then I move down and find out if there's anything that's unexplained or anything that's contradictory. So it is a very valid thing. So he's asking us to try to make the young Earth, to try to look at the young Earth model and see if it works, right? Which is what we pretty much do on this channel, right? I think.
I would say it's pretty much what scientists have been doing for like
forever,
for a while. Like of course they stopped at some point because they realized, okay, this just doesn't work. Like the underlying assumptions don't work.
Right.
So I think I don't think it's as prevalent to study, if you want to call it that, as it was back then.
Yeah.
Because it's kind of what I was saying earlier. Like the strength of a theory isn't so much its sacrosanct status. It's more so, okay, how much of the data does it explain, and how well does it explain that data? And that means that darn, I lost my train of thought. Apologies.
That's okay. You're old. We understand you're getting old.
That's right. Your older heart.
And the mind starts to slip as you get older. So we all know that.
So Andrew, do you
boom,
walk into your bedroom or something for some reason and then forget why you went in there in the first place?
Sometimes.
Okay. Yeah.
That is a function of the structure of the human brain. And I can tell you about it sometime. I think I have in the past, but
I think you have, yeah.
Yeah, it's really cool.
I had an idea in the living room or something, and then as I start to go to the bedroom, I'll see things that remind me of other things and I'll have this internal dialogue. Then I'll get into the bedroom and forget why I came to begin with, and then sometimes I'll remember later, why I went in there. And realize there was an internal command in my brain that just kind of got obscured in the memory.
But there's another thing. If you go back into the room that you came from, where you first got the idea to go get the thing, you will immediately remember it when you go back into the environment where you first decided to go get the thing.
Especially after you sat down, gotten all comfortable, the dog's on the lap, falling asleep under a blanket on your lap, and then you remember, and you have to wake up the dog again.
Yeah, which I hate. I hate waking up the cats. And they give me that look like I've really harmed them deeply. Yeah,
they're wondering whether they should have you put down or not.
Yeah, exactly. Yeah. Okay. So
before we keep going, I'd like to apologize to Lena for something I was thinking just now, because she was talking about Slimy or someone like that, and I thought she was referring to me, but I guess it was more Sal. So I apologize.
Ooh, so Lena has an opinion about Sal too.
I guess so.
Now Lena's a Christian, right?
I think so.
Yeah. Okay. Interesting. But one of the more open-minded ones who actually gets treated better by atheists than any of those Christians treat her.
Oh yeah,
they have been really harsh. So. But I wasn't sure.
It'll be a happy day when she decides to come in here and actually engage us in conversation. She says that her accent is what gets in the way, and but like I work with developers who are contractors out of Ukraine.
Yeah.
And some have good accents, some have little thicker accents than others, but we can usually figure out how to talk about code and systems and stuff. So I would think we could talk to Lena.
Yeah. I had a problem in a recent contract because I had terrible problems with accents, particularly East Indian. I had terrible problems with what is the other one? There's another one that's really bad, but Eastern European, because of my ancestry, and my uncles sort of had that tone and style in their voice, and so did my grandma and grandpa. That usually isn't a problem for me. The worst problem I had is when I worked with an Italian who moved to the Ukraine, and he was like a real fast talker, and I couldn't understand a word he said. Not a word. Not a damn word. But yeah, I usually don't have a problem with it. So I don't think it's as bad as she thinks it is. So okay, so I'm on 16, and for some reason I flagged this slide molecular phylogeny. This is where he gets into the phylogeny thing, where he shows you blast and the alignments and mega, I think, and starts talking a little bit about Erica. So
and with Erica.
Ooh, I called that, didn't I?
And let's see.
Yes, you did call it.
I found it.
Yeah, but then he doesn't really get started till here.
Oh.
Okay, and then let's see. Published first. Who do we have here? Petrie. So okay. So is protein classification necessary towards alternative approaches to function annotation?
Now, I mentioned earlier about some of the issues with looking for a universal common ancestor among proteins and genes, and it's related to this paper is protein classification necessary by Honig and Petrie, and also Koonin and Wolf. They're talking about the lack of universal common ancestor among proteins and genes. Now, that's not the same thing as the universal common ancestor of organisms. And what I found in like the last decade or so, that the idea of a universal common ancestor in terms of organisms has been kind of, it's ambiguous now. They're talking about a universal common ancestral population. So when you're talking about the domains of life, that's now, even by evolutionary literature, they're like, no, it's not from the same single cell. Maybe it was a pool of cells. I mean, this idea has been floating around since the time of Carl Woese and Doolittle.
Yes. Okay. So this idea of it being a population or a bush has been around among population geneticists since population genetics came about. And the idea of a universal common ancestor came about with eukaryotes, where people just couldn't really imagine more than one mitochondria invading a cell, right? But actually, when you draw out all the different things that happened to make the transition from prokaryotes to eukaryotes, you're ending up with population. I mean, you best view a gigantic population, and at some point there may have been. I mean, if you did a coalescence analysis, you may go back to a single ancestor for some of these structures, but you won't find it for all of the structures. There's going to be all of this all of this horizontal transfer, this merging, all of that stuff. And when you go back into progenotes, which basically did their heredity by being a bag of content, right? They had a certain content, and that was heredity. When they divided the content between them, that sort of thing, with slight differences. So the idea of the universal single cell common ancestor has been disfavored for a much longer than is on Cell's radar here. He kind of implies that we just came up with this idea.
Well, it also seems to me, and I think Immutable said this in the chat, that like common ancestor doesn't really necessarily refer to like a single individual.
Right, it's a population.
Yeah. Right, it's a population. It's more a species or population. Yeah.
Yeah, and then the other thing, so he draws a line between that and a universal common ancestor for proteins. And now the first time that he ever talked about this was on one of our Saturday Night at the Atheist, where he was like, okay, you're admitting that there was no universal common ancestor for proteins. And I said, yeah, it's pretty much understood. Nobody would ever think that all proteins evolved from one protein, right?
Right, that doesn't...
I mean, yeah, if you know anything about molecular biology, you will never have formed that belief.
Yeah, I don't think I've ever heard of anybody saying that other than Sal.
But he makes a really big deal out of this, and he has a bunch of slides that come up later. Let's see here.
Because it seems like if the original protocell had only one protein, it wouldn't really function, would it?
Right. Yeah. It had to have a group of massive, probably many more times than what we have now, kinds of proteins all at once. And they eventually started to select out. So only, once we went to a DNA replicate, then we ended up with far fewer proteins than were probably present in some of these early organisms. So, but I'm trying to find where let's see, problems with Cell, Jensen and Carter, horizontal transfer, eukaryotes again. Why did I not? Oh, unwittingly. Okay. So we'll go back to where we were, but I wanted to show that he put this slide up that briefly. It said evolutionary biologists, you see, implicitly, reluctantly, perhaps unwittingly, accept the orchard model for major protein families, but not for organisms. So he thinks that was a hard pill for us to swallow, which indicates to me that he does not get at all what we're talking about or what's happening. That means it's totally gone over his head, because he thinks this is like really significant. And he talks about it several times during this broadcast. And I think if Cell is to learn something, he needs to learn that is not the case. Now, let me go back to my bookmark, Universal Common Ancestor. Okay. So he's talking about these two papers, and I have reviewed one of them. And I'm going to have to try and get these papers into the description. But one of them is, let's see, it was by Wolf and Yaki. Who's the first author? Where is he here? Okay, he's got Petrie. So Petrie has a thing on protein. Is protein classification necessary? And how you guys okay with digging in?
Sure.
Fire away, sir. Fire away.
Okay. So stop screen. So I'm going to move away for just a minute. Share screen. And see if I can find the one I want. And I believe it's this one right here. And I have not looked at this paper to see why he included it. Okay. Hopefully you guys could read this. Can you? Or do I need to get much bigger?
I will full screen my window.
Let me see if I can get it to the point where people can read it.
Yeah, it's readable to me.
Okay. So I got to move over a little bit. Okay. So what do we got here? So this is, what is this? This is a current opinion. So this is an opinion piece by Donald Petrie and Barry Honig. Current non-redundant protein sequence database contains over 7 million entries and a number of individual functional domains. The vast quantity of data associated with these proteins poses enormous challenges to any attempt at functional annotation. And then they go ahead, I did review this. Classification of proteins into sequences and structural groups is a widely used approach, right? So they come up with a way to create phylogenies or families of proteins. But the problem is that in this article, we question such strategies. We describe how multifunctionality and structural diversity of even closely related proteins confounds efforts to assign function on the basis of sequence or similarity. So what they're saying is to abandon the rigidity of the approach of classifying proteins, because many proteins, like you'll have a family of proteins and one or two or some subset of that family will share a domain from a totally unrelated family. So all of a sudden you've got a bush again, right? So you got a problem because of the way proteins recombine, duplicate, exon shuffling, several copy errors. There's all sorts of shuffling going on, so it's very hard to see these families as being that particularly clustered, right? So now we're looking at a bush. Okay.
Are we using function in the colloquial sense, or is there some special connotation of function that we need to know about as it applies to science like this?
It would be in the biochemical sense, like you'll have one protein that maybe attaches to a piece of DNA and does some regulation, but then you'll find out that it also works as a signal molecule in another pathway that's totally unrelated. And that's what they mean by function. So they're talking about these biochemical pathways. That would be the function,
basically,
right?
Scientists think they can assign or determine functions based on structure, but there's really so many exceptions that it's not really a good rule to think that you can do that.
Right. Yeah, there's just too many crossovers, and it's too much of a bush. And these guys have the opinion that we should abandon that approach, but then I am of the opinion that approach will give us some information, right? We have to manage the fact that there's going to be a lot of crossing over, and we're going to have like this nice line of circles with a hierarchy in a family, and we're going to have a lot of arrows coming from other places, right?
So there'll be like a range of probabilities, and some of them, there'll be so many. Yeah, like you'll say this structure does this, but there'll be so many exceptions that it's hard to say that we know what that physical assembly of proteins actually does.
Yeah. Yep, that would be fair. So Kunin, 2008. Why do I have two of them? Oh my goodness. Where is Kunin 2008? Oh, Kunin 2008. Okay, let me show this one. So the other paper he mentioned shortly after is Kunin 2008. And he claims that they had a problem with that. They were surprised or had a problem with common ancestry for proteins, right?
Right.
So I went and looked at the paper and tried to figure out what he's talking about. It's like, what are you talking about, dude? I don't know what you're talking about. So I had to figure it out. So I went and searched for common, and I found, I sure wish this was cleaner, guys. I apologize. It takes me a while to get this shit right.
No problem.
Because when you move it over, it gives you white space instead of showing you the text. It would be cool. So if we look at common, okay, the only time it mentions common ancestry right here is it said genes that evolved from the same ancestral gene and the common ancestor of compared genomes. So let's see, what is he talking about? Genes that evolved from, okay, so they're talking about families. They're never claiming that all genes evolved from a common ancestor. They're claiming that they evolved from families, right? So he never mentions it in this paper at all. So somehow that paper was misread. Okay, more precisely, full count provides for the possibility to identify sets of orthologs, genes that evolved from the same gene in the common ancestor of the compared genomes. And when I looked everywhere else, I couldn't find anything in this paper. And this is Kuna in 2008. Common mode leading, common, so no, I couldn't find it anywhere else. Here we go. Last universal comparative genome, minimal gene sets, and the last universal common ancestor, but it's not about proteins. So this paper on Kuhn that he mentions does not have the import that he says it does. And that is my point here, right? You
What
follow
about that
that?
other Kuhn? What about that other Kuhn that says how many whatevers make a cell?
How many?
Yeah, it was just above that one Kuhn that you noted.
There's only one Kuhn, I think.
I thought I saw two.
2 Kuhn's? I have 20 raccoons. Maybe you're confused.
That's a significant number of raccoons.
Yeah.
And they're all overfed.
I think we only have 1 Kuhn though. So, what did he say about cells? Can you remember?
No, no, I don't know what he said about cells.
Okay. But we'll keep an eye out for that because you brought it up. Oh, I had to double check to make sure I clicked the audio thing. So we're sharing. We're back to the thing. Wait a minute. I got to pick something. I get it. I'm good at this. I'm on the ball, man. Okay, let's see what he has to say. Does anybody have anything else on that? Is that understood that he probably that paper does not provide the evidence that he is looking for?
It seems like it, and this supports my continuing doubtfulness about his reading comprehension, not because of intelligence, but because of
worldview.
What he's looking for, what he's trying to get to.
Yep. Okay.
Now, I mentioned earlier about some of the issues with looking for a universal common ancestor among proteins and genes, and it's related to this paper's protein classification necessary by Honig and Petrie, and also Koonin and Wolf. They're talking about the lack of universal common ancestor among proteins and genes. Now, that's not the same thing.
Okay, there it is right there. I didn't find that in the paper at all, so I don't know what he's talking about. He's going to have to come back and tell us what he's talking about. If he does have a point, I invite him to email me or better yet, because he won't talk to me, talk to one of you guys. So
he pointed to authors and not their papers.
He's got the paper up here on the screen.
Okay.
So yeah,
but that's the one that doesn't.
That's the one we just looked at.
Yeah, it doesn't support him at all. Yeah, that is a continuing problem among young Earth creationists, is that once you read their papers, they don't support the point they think that they're getting support from.
Yeah, and I'm gonna be generous, and I'm gonna say sell. Maybe I didn't read the whole paper. Maybe you found something that I didn't see because I didn't have time to read the whole paper. So let us know if we got you wrong on this.
Fair enough.
Through Andrew, because he won't talk to me because he hates me. So
okay.
No, he doesn't hate me. He's aggravated by me for some reason. Maybe it's because of this. I kind of dig in a little bit. Maybe Sal doesn't like that. I don't know. It's an idea. Oh, there I go. Ad hom. Sorry, you're supposed to correct me.
Sorry. Hey,
Sorry.
you corrected me before I could hit the mute button and unmute myself. So good job there.
Like you could have removed me from the studio. So
you have to donate money to a charity now.
Okay, I just Venmoed $1 to my favorite charity.
Okay, good.
I think it was buffaloes being eaten by Komodo dragons. It's a real problem.
Okay.
Yep, and I do have an image.
Oh, cool. Nice.
For Snata tonight. Okay, moving on.
The universal common ancestor of organisms. And what I found in like the last decade or so that the idea of a universal common ancestor in terms of organisms has been kind of, it's ambiguous now. They're talking about a universal common ancestral population. So when you're talking about the
I'm repeating myself, ain't I?
Seems
Or
like,
are we? Yeah, okay, universal common ancestor.
yeah, 'cause
Okay.
I remember ambiguous.
Yeah. So there's a little more of this for anybody listening. Things get juicier. We have some juicy things to get into later on, so that are pretty good. Okay, Dennis R. Venema, professor of biology at Trinity Western. Trinity Western is a Bible college, and Venema was in the in that trial. Where was that? I can't remember the trial that they. Yeah, that one. Yeah, I believe he was one of the witnesses, wasn't he? Anyway, so the next thing that they get into is how they went after the Nyelinase bacteria. Okay, Ono said that the Nyelinase mutation was a result of a frame shift, and he said this back in. Well, let's play a little bit.
Also, Dennis Venema in 2016 advocated, and in fact, this particular claim, it was his favorite claim about protein evolution. And I said, okay, this is going to be a nice big fish if we could show that the basis of what they're promoting is wrong. And this will lead to why I was interested in phylogenetics. I might be.
I don't actually know Venema. Do you know? Is he a creationist or what is he?
I don't know.
I thought he was, but I can't remember.
I've never heard of him up to this point.
I saw the Dover
he has a
video
book.
a long time ago, but I don't remember the name.
Yeah, he has a book. And he works at a Bible college. Yeah, go ahead. Look it up for us.
Able to even see if I have it here.
Wow.
Yeah, I have the mega program going. So hang on.
Okay.
So I'll just proceed here. And he said evidence of evolution and how new proteins can evolve is based on this.
Let's look at that.
New proteins can evolve is based on.
Okay. There's an article, National Center for Science Education. Okay. Basically, this article talks about how new they push back on this whole idea that new proteins needed God to be created. And I can also link that in the description. But I thought there's just some information in this article. I do have it up somewhere, but I don't think we need to look at it. So anyway, they show you how proteins can evolve without God's help. Are they?
So this is the proteins to praise Jesus view of
no, this is the opposite.
Biology.
This is the opposite. This is a actual science article that tells you no, we don't need God's help to create proteins, de novo proteins.
So this is Creation Evolution Journal. I thought.
Yeah, but anyway, so to abbreviate things for the next hour, I think, or half an hour or 45 minutes, you guys get into Mega, and he shows you where the Nyelinase, basically where Ono was wrong. And Ono was probably wrong about the frame shift, but Ono wrote that paper in 1985, and it was a damn good guess, and it was, a possibility. But then they found remnants of this Nyelinase ability, or the things that are required to enzymatically break down nylon. They found them in other bacteria that exist, that go way back, right?
Right.
So basically, they showed Ono was wrong on that particular point, because that was a famous, and I think they used that in the trial as evidence. So Cell caught a big fish and went ahead and Cell actually did a paper on that, I think, with Sanford and one other gentleman, and basically tore that apart. But I have not looked into that. And I'll bet you $1 if I look into it, it's not going to be exactly the way Cell is describing it. But I will look into it, and we will get there eventually. But so anyway, so then Moina arrives at 1:33, and you're kind of like out of the talk, unless there's anything else that you wanted to address that you were talking to Cell about.
I guess the only thing would be that clip where he says there's powerful evidence for common descent, but other than that, yeah, we didn't talk much after that. It was mostly.
Yeah, I'm not sure. I'm not sure if I have that one. Okay, new protein without God's help. And then there's this dude. This guy.
This
Oh shit.
it's based on sorry.
He mentions that guy later,
I'll
but
talk about it specifically.
he's a professor. His professor, I think, but he's holding a Bible.
Yeah, I keep wanting to get onto you for flashing between images, but then I realized that's
not me.
That's Sal doing it.
That's Sal doing it, right? Okay, so it was this guy. I think he said he was his professor or something, but I'm not sure. And he worked with Kunin. And I don't know if that's the guy or not, or if I got something wrong here. Well, now that I fucking said it, I
based
better play
on this. Let me see if I could find it. It's based on. Sorry.
It must be in another part, unfortunately.
I'll talk about it specifically. I have the paper in mind. But both Ken Miller and Dennis Venemus cited a paper by Ono, and it was also cited in 1985 by the NCSE, and they wrote this whole big thing, the new proteins without God's help.
Okay, so Venemus.
The creationists will argue for the improbability of evolutions. So we wrote this paper. It got rejected by several editors.
Yep.
We asked the editors to release, if it'd be okay if we could release the rejection letter. They would not. And there are a number of maybe good or bad reasons for that, but that's just the way it was.
Are you prevented from releasing your own rejection letters? I don't think so.
That's what I was going to ask. Why would they have any control over that?
They don't have any control over that. They sent you a letter. You have every right to publish that letter.
Yeah, so if Sal's against that, then we are in agreement. I think you should be able to. I don't know what legal hold they could have over you.
No, they couldn't. They couldn't do anything. That's not copyrighted material.
Paper, then any agreement with them not to disclose any correspondence is null and void because they're not gonna print your paper.
Yeah, if you send me something in email, unless you've gone to a lot of trouble to copyright that mail, you have absolutely nothing to say about me showing it to anybody.
Yeah.
Which is, there would be so many lawsuits, wouldn't there?
So if Sal is outraged by that, then I'm outraged as well.
Yeah, I know that would be outrageous if they. Okay, here we go. Who has design on students' minds? So he talks about this guy again.
Phylogenetics. That's one of the scientists I've worked with. He's a young Earth creationist. He's a adjunct professor of at Vanderbilt School of Medicine and also associate professor at Lipscomb University in biochemistry. So there he is. That's Joe DeWeese. He published in the prestigious scientific journal Nature. Let's see if maybe a little bit of bragging rights here. I actually was in the prestigious scientific journal Nature in 2005, April 27th, just for the reader's benefit. The cover story, who has designs on your student's mind, you'll see me in that cover story. So just to say, a young Earth creationist got published in a major scientific journal on the topoisomerase protein. And then both of us got published on this in 2019. Unfortunately, it had to be April 1st. It's like, no, that wasn't an April Fool's joke. That was for real. So this is the paper on
Dannekins, real quick.
That was in question. They said this is this was cited for.
So that chemical or discussion, that biochemistry paper had nothing to do with biological evolution. It looks like it was talking about treatment of something.
Yeah, that one was about topoisomerase that they did, that they published. And I haven't pulled those papers up today, but I have them out somewhere on the drive. And that was where he published with DeWeese, I guess, in Sanford and stuff. Let's take a look at that and see if I can get that paper back up. Okay, variable C domain. Okay, so cell Cordova, DeWeese. Oh, I guess it isn't Sanford. I thought it was Sanford. So nope. him and DeWeese. Human type 2 topoisomerase as a functionally relevant therapeutic agent target. And I don't understand what the paper does for him, to tell you the truth. I don't know. But I know he gets into topoisomerase here later on, and that's kind of where we're headed. So
19, that was in question. They said it happened by frame shift.
yeah. Anyway, so the frame shift thing was probably wrong, but we'd have to look into it and find out exactly how he got it wrong and what the nylon breakdown ability came from, and the chances that it came from other enzymes and things that were present for a long time. It sort of supports evolution, because protein evolution, that's how it happens. There's all of these different motifs that each have enzymatic function, and when you recombine them in different ways, you create brand new function, brand new proteins. So
yeah,
I think
maybe we should we, I guess, go into it after we've looked into it some more.
Yeah, we'll go into Coonan after that particular thing, and see what the whole controversy is about. Okay. So now we're over here where Moina arrives, and I don't really care about that. Immutable arrives.
Oh, so you don't have a clip about any other common ancestry stuff?
I couldn't find it, but go ahead and tell us what Cell said.
Well, along the lines that I remember, he was basically saying that well, yeah, he actually said, okay, this nested hierarchy, it does exist, and we actually agreed that the analogy that SFT puts up, for instance, like he didn't mention SFT directly, I think, but like the analogy with cars and trucks and things like that, he said that's not a very good analogy. That they need to put up more than that. So we actually agreed on that, and the fact that this nested hierarchy is powerful evidence for common descent. I think he just thinks that some parts have to be designed, which of course I disagree with, but that's kind of where we parted on that. And that's kind of the chat, and then Immutable came in, of course.
Well, and then he hinted that he was going to tell us later about his idea about why that phylogeny looked like that, why it looked like common descent. And to the best of my knowledge, his idea is that God created it that way, and he created chimpanzees so that we don't have to do medical tests on human beings, because they are so similar to us that we can do medical tests on them instead, which I consider
reveals horrible ethics.
I think, animal ethics anyway. I think so. But he said that famously on a number of SFT streams and Natox's streams and stuff like that. And if anybody doubts that, I could go find clips. So anyway, but that's what. Go ahead.
Oh, or no, go ahead. You say what you were going to say, and then I'll say my thing.
I totally forgot what I was going to say, Andrew.
Oh, darn it. I'm sorry. I was just going to ask if any word on Erica or any news on anyone else.
No, she hasn't responded.
Oh, well.
She might be actually going out having fun Saturday things.
Nobody loves us.
Yeah,
nobody cares. She'll probably show up on Saturday night at the Atheist tonight.
In that case, should we let Danny in and like talk about the whole mathematics thing last night that I had?
With Dan, is that what he wants to talk about?
Or no, I just think like what would Danny know how like the philosophy of mathematics and how it's not empirical, like not purely empirical?
Yeah, he probably would, but
or up to you guys, whatever you want to do. Speed.
I'm not letting Danny into this stream right now.
Okay.
Yeah, so I don't. Yeah, that would be another topic. And for the weather report, maybe on Monday, Danny can come in and talk to you about the empirical notion of mathematics.
Okay, sure.
That sort of thing. Actually, like I say, I would push back on that a little bit, because I think it is at a very basic level empirically derived, but not through empiricism in the sense that empiricism, right? Through a different sense. So anyway, what's that big dog's name? You got Big Dog?
Oh, this is my little guy. This is Gibbs.
Gibbs.
Let me see if I can show him off. Damn it!
Can you show Gibbs?
Gibbs the energy.
Okay, Cam, Mike. Should be easy, right?
Should be easy.
Supposed to be easy.
You just turn your cam on. That's all.
Well, it's on. It's just not showing.
Oh yeah, they do
Oh,
that.
start cam. Duh. Okay. Whoa. He just jumped down. He knows I'm about to show him off.
Okay.
Hello world. You know what I look like now?
Yeah, you're excellent.
Hell yeah.
You look like Santa.
I kind of do.
You kind of do. Okay. Now, if you get them again, let us know. Anyway, okay. So now we get to the part that I like, and we get to zinc fingers. Okay.
Problem with macroevolution of, like, say, okay, just to simplify it, this collagen is not going to evolve to be one of these because it's going to.
Okay, he's showing a collagen protein and a zinc finger protein, topoisomerase, I believe, or maybe, I don't know, TF. What is it? 3A. I'm not sure which one he's showing here, but anyway, no one ever claimed that collagen was going to evolve into topoisomerase. So there again, you have this idea that all proteins had to evolve from one protein or common ancestor. And again, he is very misled on this stuff. So, and this leads us to zinc fingers, which are, let me play a little bit.
Going to break
here.
its collagen pattern. Or if we tried to.
Did somebody say something? Oh, there he is.
Dog.
There he is.
Oh,
That's my
he is
picture.
adorable.
Oh my God, it's little.
He's
What is that?
He's a cute little guy. He's a coward, but I love him anyway.
Is he a mini
He's very cute,
Doberman?
Scott.
He's a Chiweenie. He's a cross between a Chihuahua and a weenie dog, a dachshund.
A Chiweenie.
Although most Chiweenies look more like dachshunds, I think, but this guy was just, he was the run of the litter and he was just a little different.
Okay.
He's afraid of things like if I happen to set an empty paper sack next to me on the couch, he won't get up on my lap because he's afraid of the paper sack. So
it looks like there's a Chiweenie in chat too. I just displayed one of his quotes. We have a Chiweenie in the chat.
anyway, that's my little guy here. I'm gonna go ahead and turn off the camera again.
Okay, so Danny is probably related to your dog, Chiweenie. So, okay.
Kind of reminds me of one of my dogs.
So what the fuck is Danny saying is gibberish now? What is he talking about?
I have no idea.
Oh, about my empiricism. Yeah. Okay. Yeah. Danny thinks it's gibberish because he doesn't understand it. Good enough, Danny. We know you're a Chiweenie. Okay. So let's go on.
To evolve this to a collagen, this zinc finger to a collagen, it would break. And by way of extension, it'd be very hard to imagine what the common ancestor would be that could be functional because you don't have a collagen and a zinc finger ancestor that can do both or something similar. It's just not going to work. And so I asked Dr. Dan, CRISPR-Cas, both immutable and speed of sound, and they said, yeah, they're cool with the idea of independent origins for the proteins. I said, okay, at least we have some sort of agreement that there's
not just cool with it, but it's the only fucking way it could have happened, Sel. And I told you that over and over again. I'm not just cool with it. It's all about evolution. It isn't something that I'm just, tipping my hat to. It's actually the basis of all of this molecular mess that has created evolution. So
shouldn't it have something to do with like exaptation, for instance?
What's that? Oh, you mean proteins?
Yeah, like or can proteins be like co-opted to do like would that work here? I don't know. I'm not an expert.
Oh, that and about 50 other mechanisms. I mean, it is such a mess. It's like, you got a train yard and you put together a bunch of boxcars. You have all that train yard DNA out there, or you could call it workshop DNA, or you have a workshop that's just stuffed with remnants of all sorts of old machines and parts. And you go out there and you grab a little of this, and you grab a little of that, you slam it together and you get the job done, right? That's what's going on. I mean, that is so obviously what's going on, and that folds into neutral evolution and all of these other things. Okay, so I can't invent definitions. He says
okay,
I
Steve,
can't.
Let's not.
Oh, I'm distracted. Okay, I'm sorry. I'm sorry, Andrew.
I pat you on the head.
I lost it for a moment there. The way should I remove him from chat so he doesn't distract me?
Yeah. If you do that, I'm gonna leave the stream. I'm sorry.
Okay,
I don't condone such behavior.
I can't block Danny.
No,
the Chiweenie. Can I call him a Chiweenie?
If you want to insult him, sure. Just don't take any moderation actions.
Okay. But I want everybody to note that Danny doesn't have a wrench, so if anybody else wanted to remove Danny.
You know, I can't really support the idea that Chiweenie is an insult.
Okay, so I'll just
Chiweenies
call them.
are cute little affectionate fuzzy animals. They're just a little bit neurotic, that's all.
Yeah, well, Danny is exactly like that, so we do love Danny. So we all love Danny.
Okay, well, that's good. That's progress then. So please note, Danny, we do love you.
We have a soft spot for Danny. That's why we tolerate him. And where nobody else would. Okay.
Kind of an orchard-ish model. I mean, the creationists will say these are created. Andy, go ahead.
Yeah, there's something I should add too. So like what you're mentioning, the different colors that you have for this, I'm assuming it's an amino acid sequence, right? For either collagen or zinc finger, right?
Right.
So something like basic enzymology for some people is that, look, this phenomenon shows up in basically all proteins. All proteins essentially require a handful of amino acids that need to have a specific geometry relative to one another in order to create an active site. But what I should add to that is that while your point is valid, Sal, I think if you're looking in terms of like common ancestry or things like that, or if what you're trying to do is use common ancestry, what you might be interested in doing is instead of observing just the active sites, you're familiar like with various folds that occur in proteins, right? So like alpha helix, beta sheet, things like that, and the higher order structures that they form.
Oh yes, yes.
Right. So what I propose, right, as a way of kind of thinking about that is first the question is, if you're making a protein, why bother having these higher order structures that proteins have? And to me, the answer is because certain properties that those residues adopt, like alpha helices or beta sheets, lend themselves to being more likely to be in some environments rather than others. So sometimes alpha helices are very, very polar, based upon the way that the side chains of the amino acids have to interact. And so you would expect it to be sometimes facing out of the membrane rather than facing in, where beta sheets tend to be more nonpolar in structure.
Okay, and I want to mention that there are 10 nonpolar amino acids out of 20, and there are basically 4 different kinds of amino acids. So statistically, it is not very hard to do a random throw of the dice and end up with an alpha helix with the nonpolar edges sticking out, so that it would fit into a lipid membrane, that sort of thing. So, I mean, that is not surprising at all. Now, what is surprising to Cell on the topoisomerase, maybe I got to go a little bit further here. Let me see, looking at problems. We'll try it here. We'll see what we got.
Appreciate those sentiments.
Oh, shit. No, I went too far. Okay, so one of the things Cell says is for that topoisomerase, or up here to phylogeny not proof, monotic rival, immutable. Where were we? How did I lose it? He shows the topoisomerase. Okay, I think. Let me see here. Okay, the zinc finger proteins, and he shows the fact that these things are lined up like that, and he talks about how impossible that would be to happen randomly. That we have histidines here, and we have histidines here, and here we have cysteines in the red, and those things line up with a certain amount of space between them. Well, here's the thing: once you have one of these, an easy accident will end up with 2 of them that are pretty similar, right? A duplication or a recombination. Mm-hmm. And from there on, all hell breaks loose. And these proteins will keep duplicating these things over and over again. In fact, between you and I, if we looked at our topoisomerase, I may have 22 and you'll have 21. So, I mean, it happens very commonly, particularly in primates. So it's like, and famously, SFT makes a big deal out of all these duplicates that chimpanzees have and other apes have that we don't. And there's a reason for that. It's because it's a very easy problem to occur when duplicating genomes. And as it turns out, the problem, and I've got a lot of papers that I could go through, probably not this afternoon, that show you how these incredible patterns, like there's like barrels and what else? There's just all of these incredible patterns that are formed, like what are they? They're called propellers. Propellers form due to this duplication, which is ubiquitous in the genome. So once you end up with one of these, and the other thing about a zinc finger is all you need is 2 serines separated by not even, well, I think they're separated by 1 amino acid, maybe. I'm not sure if they even need to be. And then you need either two cysteines, and then another 2 cysteines on the right, or 2 histidines. It doesn't matter, and they'll form a zinc finger. Now, zinc finger proteins are present in 3% of our proteins, and that's just the known proteins. 3% of them, which amounts to about 1,000 proteins, have zinc fingers, because it is very common to get 2 cysteines in a row, and then either another 2 cysteines or another 2 histidines out there, and you got a zinc finger. You got 1 finger, right? And Cell makes this. He makes it look like it's extremely improbable, and that lining up is not improbable at all. That happens very easily. Okay, so the other thing is that what zinc fingers do, this stuff in the middle is sort of the knuckle, right? I don't know if you can see my cursor here. The stuff in the middle is called the knuckle, and the knuckle is capable of attaching to DNA and a whole lot of other molecules, to tell you the truth. Zinc fingers do a whole lot of different functions. One of the things they do is they attach to a particular string of DNA, and it depends on what's in the middle here as to what it will attach to. Now, it doesn't have to be a perfect fit at all. It just has to, you have to have certain nonpolar amino acids and certain basically acidic or plus or minus charged amino acids, or some other polarity, and it will attach to some degree. It may not be a perfect fit. And then if you give it a little time for mutations, the fit may get more and more perfect. And sometimes they become very specific. Now, there's a whole bunch of zinc fingers mentioned here. And later on, he uses an example of transcription factor 3A, which is TFIIIA. And it was first found, I think it was found in a mouse embryo. And that was I think it was the first zinc finger transcription factor ever found. And it has about 20 or 17 or 20 of these zinc fingers. Only 3 of them actually attach to the ribosome 5S particle, which helps you create ribosomes. And so it only takes 3 of them. So not all 17 even work. So it's kind of a work in progress. So again, he's making it look like something extremely improbable, or something that is probable is extremely improbable. And he does this, and he believes this, because I don't think he's looked into the papers that tell you how these zinc fingers actually work and how ubiquitous they are in biology. So that was and I have a whole bunch of diagrams, and I have a PowerPoint presentation, and I could do a more technical stream on that and try to explain all of that. But let it, we can leave it as it is nowhere near as improbable as Cell thinks it is. And that was kind of his, when he said that phylogenies were strong evidence for common design, he then went on and said, but this takes a miracle. So this is Cell's whole body of work, is these zinc fingers and topoisomerase and how it takes a miracle to create them. And it topples as soon as you start digging into it. He has nothing. So anyway, what do you think of that, you two? I diatribed.
That was quite an informative diatribe.
Okay. So I made sense? You're saying I made some sense, or was that gibberish?
Well, it made as much sense as I could make out of it without
Okay.
knowing all the words.
Yeah, I could show you in diagrams and I could explain it, and we probably should do a video on that at some point, because this crap keeps coming out of his streams over and over again, where he's talking about this improbability of these things developing, and when you look into it, is nowhere near as improbable as he suggests. So, which I already said. No, I'm fucking repeating myself. So, let's see, collagen evolving from drink. Double knockout Brenda. Okay, here unwittingly. So double knockout Brenda.
Experimental evidence that there is a level of robustness. Brenda Andrews did experiments where she was knocking out genes, and she was actually shocked at her results. These single knockout experiments, she couldn't believe how many single knockouts she could accomplish in the yeast. Granted, that's not
not exactly
but she was still like, wow, hardly any change in reproductive fitness. Once she started doing double knockouts, it became evident that you can't, it's like spare tires. One can act like a spare tire if you knock out one, but you can't knock out both. And so you do cite an interesting property that there could be mutations to the system that are not immediately detected, and that also leads to a whole nother topic of deterioration. Then,
so they're talking about genetic entropy and how you can knock out one gene without any problem. And okay, so genetic entropy fails for a number of reasons. That's only one of their points in genetic entropy, is that you can knock out one gene, but what happens if you knock out two? So Brenda Andrews did a bunch of papers on yeast where she did knockout experiments, and she actually talks about not 2 knockouts, but 3 knockouts, because 3 knockouts tend to really screw things up, right? So if you end up with 3 knockouts, what happens is you have a deleterious mutation by the time you get 3, and then you would have a problem. And if everybody in the population already had those first 2 knockouts and you did the third one, you'd end up with an extinct population, unless there were some outliers that didn't have those knockouts, which if you listen to them, it takes it takes many thousands of generations to completely take a neutral mutation and have it go to the entire population. Takes a whole, there's something called Muller's ratchet, I think it is, and a few other, the waiting time problem and a few other things they've talked about. But if the mutation is truly neutral, it has to spread through the population randomly, statistically, and the chances of it completing the entire population are very slim. So you're always going to have these outliers that don't have that knockout, and that's why genetic entropy does not work. It is possible that you'd end up with a very, like if those 3 genes that were dependent on each other were knocked out by random mutation, that's a 1 over 3.2 billion possibility 3 times. So 1 over 3.2 billion cubed is the likelihood of that happening to all 3 of those genes. Then it would have to fix the entire population, or basically the 2 knockouts would fix the entire population. The third knockout would come along and none of those guys would reproduce. So if the third knockout, if none of them reproduced, then how could it fix in the population? So genetic entropy is down for the count. That's, 3 strikes and they're out because it could not fix, right?
Right.
And even if it did, there would still be outliers that didn't have this problem. I mean, even if every single organism out of a population, or suppose you had an extreme bottleneck and they happened to all get that third mutation at the same time, second or third mutation, it's just very unlikely that any of that would be a problem for evolution. So,
right.
So that's the point. So double knockout Brenda, I went and looked at her papers, and I looked for the word surprised and shocked in a lot of her papers, and I didn't find any of that anywhere. So I don't know. Maybe
other more mild words which are equivalent to the word shocked in science lingo.
I would have to get a thesaurus out and write a program to search all the papers and try every combination. I don't know.
Yeah, shocked equal it raised my eyebrow or something like that.
Yeah, I don't know. I'll have to look. Okay, and then he goes on.
and where I have gotten in some sharp disagreements with evolutionary biologists, saying that you can't use this to say that it evolved naturally. And it was in this talk. This is a slideshow from the video on this, where this is some extraneous stuff here. Let me get to the heart of the issue. Okay, so this is the mega alignment between humans and bacteria. And okay, this is one thing of interest. The bacteria, what it doesn't have is a sequence like this here. This is a localization sequence. This is it's called a bipartite nuclear localization sequence. So I highlighted in red here where it resides on the human, and it doesn't, the bacteria have amino acids there, but it's not this sequence highlighted in red. What's the importance of this sequence? This sequence resides somewhere physically out here on the topoisomerase. This is the fold again. That's Dr. James Carter, not Dr. Robert Carter. That localization sequence tells the it tells if the topoisomerase is somewhere out here after it's been translated from the mRNA, it tells it to go inside. It needs the sequence to go inside the the nucleus. So this sequence is very important. If we suddenly have a eukaryote that has nuclear membranes and a nucleus, it needs a way of having the proteins go from the cytoplasm back into the nucleus. And when Dr. Dan appeared on my channel, he said, well, you need to include natural selection in your model, Sal. He did see I'm inferring from his comments that he could see the challenge of evolving that localization sequence. And again, just for the reader's sake, this is the localization sequence right there, to evolve it by random mutation. And he said, you need to include natural selection in your calculation. Otherwise, I'm not interested. I didn't have a chance to respond because I saw his comment kind of only after the stream was over. And you know how it is in the live stream. I wanted to say, well, natural selection would select against this because if you get it half right, the creature's dead already. It's dead on arrival. And that's only one of the critical proteins. We would have to reformat all the homologous proteins. And so that was where I said, being able to build phylogenetic reconstructions is illegitimate to be able to point out these mechanistic barriers. So it's like, yeah, you have these nested hierarchies, but this is proof kind of in the Behe-esque sort of viewpoint that you would need fortuitous events or intelligent design. And of course, he'll say intelligent design, that even under the assumption of universal common ancestry, there would have to be these very fortuitous mutations. Otherwise, it's just not going to evolve. And go on.
One I'll say.
Okay, so the nuclear localization signal, or the nuclear import signal, basically the signal that imports in the nucleus, requires nothing like he said. This I have, and again, if we do a more technical review of this, we'll get into it. But it basically needs a few of these lysines out here, separated by an arginine, which is an R. So it needs a K, KK, and it needs an R somewhere, or an RR, and then it needs another KK, and then it imports. So this entire sequence here is not highly conserved. And I would have to do I'd have to do some comparisons, some blasts, and find out, and comparisons to find out how conserved they are. But from what I know, these localization signals are very loose. The chances of this randomly popping up on any protein are very good, which means that there are a lot of proteins being imported into the nucleus that the nucleus really doesn't need. So it's like wasted space until a pathway is found where it has some function, and then all of a sudden it'll become a thing, right? So this is the TF3A, the transcription factor that creates ribosomes. It attaches to that 5S and promotes it. And this import, basically this import signal attaches to that protein, or it's built with that protein. And then it attaches to another regulatory element. And then, this attaches to the 5S. They both get exported out of the nucleus along with the transcription factor, and it self-regulates the amount of ribosomes you have in your cell. So it became a regulatory pathway. And it's something that evolved, obviously, after eukaryotes, and probably a long time after eukaryotes happened, because it's not essential to the production of eukaryotes. It inhibits or it expresses 5S, and then it has a self-inhibitory mechanism. So anyway, I have a bunch of research on that. And again, that's highly technical. So I think that was the last thing he has. Is he is absolutely amazed at these import and export signals. And also I went back and found evidence in prokaryotes of nuclear import signals on other proteins before there was any nucleus at all. So you didn't need a nucleus. So these signals exist out in the prokaryote bacteria populations, and they're not used at all. They do something else. They have another function. So once again, he doesn't have a point. Okay. Oh, I got it all backwards. Okay. Can be relevant. So CRISPR is pushing back on this. And it doesn't surprise me. CRISPR knows more than I do. Okay, before the LS can be relevant, you need to have a mechanism for discriminate import to the nucleus. Before that, everything can get it. Okay, I'm not entirely sure what you mean by that, CRISPR. Oh, there has to be a mechanism to import it into the nucleus. Yeah, but all of that developed with eukaryotes, basically. And I'm guessing that there were very simple mechanisms that allowed import into the nucleus, or something else entirely different was going on inside the nucleus where we didn't really need certain things. I don't know. It's hard to say. But anyway, get in. You start with everything. Then okay, you start with everything, then enhanced transport with signal, then restricted importation. Okay, so everything gets in. And then eventually, okay, gotcha. I see what I did wrong. Okay. So at first, you have too many things getting in, and then eventually things get restricted as, the pathways build up. I think that's what it's
great to have biochemists and scientists and PhDs lurking around to
correct you,
inform us and correct us.
Love it. Anyway, okay. So I think that was all I had on this. And I think that was pretty much. Let's see. Did we do phylogeny is not proof? We did. We're ahead of that. Nuclear export signals, and then a lot more data. Oh! I wanted to point this out.
In their defense, not that I am eager to defend evolutionary biologists, but I want to be fair here. We have a lot more data now that they probably weren't aware of, in terms of the importance of specific sequences and their role inside the cell. We're only learning a lot. So there are probably questions that they had just assumed that would be easily solved that are not.
I don't think biologists ever were like that. I mean, we did think things were simpler before we knew about all of these, before we had. I mean, what is he saying here? Andrew went away. Andrew got bored.
Yeah, I think he's off getting a snack or something. Apparently he meant he went to Wendy's or something.
So Andrew, did you listen to that part?
Repeat it again. Sorry, I'm
just get that baconator out of your mouth and talk.
Yeah, get that zing finger out of your mouth.
In their defense, not that I am eager to defend evolutionary biologists, but I want to be fair here. We have a lot more data now that they probably weren't aware of, in terms of the importance of specific sequences and their role inside the cell. We're only learning a lot. So there are probably questions that they had just assumed that would be easily solved that are now emerging that they have to consider. And I just,
Okay, Danikins.
Yeah.
So he's saying, basically, scientists don't read articles, therefore creationists know more than them. Is that how I should take that?
Big assholes in the nucleus. Okay. Okay. Can you repeat, Scott? I'm sorry. I was watching chat.
You were just distracted by Casper's witty science there.
Yeah. And
No, I'm
calling
just
him,
saying, it sounds to me like he's saying, well, these new articles keep coming out, and biochemists don't keep up with them. Therefore, creationists are the modern scientists, and they're the backward scientists.
yeah.
Is that what I should take that?
And yeah, this is the rhetoric that Cell has developed with SFT and a bunch of others, that we're learning all these new things, and now it's obvious that creation is true because we can't solve all these problems that are popping up. And I did was search, Google search the evolution of zinc fingers, and I have thousands of papers to look at. So I don't think they've done that step. I mean, sure, new problems, new complicated structures pop up, but none of these things are a problem. And they believe that it's slam dunk proves creation.
So have you kept a tally of how many of the papers that he's referred to actually support the claim he thinks it's making?
His papers? I've never seen a paper that supports the claim that he's making at all.
Okay, because that was my impression, but I don't want to be misconstruing things. I just wanted to make sure there was another set of eyes on it.
Yeah, the only thing we got is a few one-off quotes that somebody said, "Oh God, this is surprising. How did this evolve? This is a problem," or something. Which biologists will say all of the time. You give them a few drinks, send them to a conference, put them on a plane, they're going to say shit. That's what they're paid to do. So anyway, so how unlikely it is that a zinc finger structure evolves through mutation? It's not unlikely at all, because the zinc finger, there's roughly maybe 8 to 12 different kinds of zinc fingers, and they have different motifs. And one of them is like the C2H2, which is two cysteines that are maybe separated by one amino acid, and then two histidines that are separated by one or two amino acids, and they fold up and they form 4 little forks that grab onto the zinc, and they fold the protein structure so that the knuckle is facing outward, and that's what fits into the DNA groove. So the chances of that happening randomly are like extremely high, that you would have thousands of these things all over the place that would have that function. So if they happen in a protein, you got a zinc finger, and that's why 3% of all proteins have zinc fingers. So, because it's pretty common. And the other thing is 50% of all proteins bind with some metal ion in their function, at least by one paper I read. So it's very common. And the interesting thing is that zinc atoms are very tiny, and you only need one or two or maybe a handful per protein. That's why you don't need to. You might need 40 grams of protein, but you only need a few milligrams of zinc. So that's why, because minerals are very important in our metabolism. So anyway, I'm exhausted. So,
well, you did an exhaustive takedown of this video.
Oh yeah.
Yeah, I think it's pretty much dead. Yeah, I'd love to see how Cell comes back on all this and gives me more data, or like you said, papers. Show me a paper where they show that this is absolutely impossible to have evolved. So, right. I would love to see that.
Especially the papers that say shocking.
Oh, I want those too. Yeah. Well, they do say that once in a while. So that has happened.
I mean, not the headline, in the actual paper.
Yeah. Yeah, not the abstract, not the headline, just shocked. We're shocked. So shocked. Anyway. Good enough. I think we've done it. You guys want to elaborate a little bit?
Well, I really don't have the language and background knowledge to expand on it much. other than the fact that it was very enjoyable, and I'm glad I got to share my Gibbs with the 17 people now watching.
Good, good. Andrew, are
Yeah.
you satisfied?
Yep. To be honest, it was a good chat. Some of it I didn't understand, like the zinc fingers, which is one reason why I didn't say much, because I literally know nothing on this subject.
That's because you didn't have them with gravy. Have them with gravy. You'll love the
maybe, yeah.
Well, at some point, I might feel competent to do a video on zinc fingers, and but I want to look at a whole lot more examples. I've got a bunch of
yeah,
I got a bunch of stuff. That and nylonase. But I'm not sure if I can do both, bandwidth, that sort of thing.
Mm.
So
Yeah.
I don't, let's see if I can, let's see, do I want to share that? Here we go. Okay, application window. So here are some of the different kinds of zinc fingers, motifs. Topoisomerase is the C2H2, which is on top here. I suppose that's completely unreadable.
Well,
Okay.
I can make it out, but these don't look like fingers. What's finger-like about them? Because they're straight. Well, not even straight.
You see that little red ball right there? Oh shit! It's not showing my cursor. Okay, there's a red. Okay, in the top one, C2H2 like.
I see a little orange ball. Is that what you mean?
Orange ball. Okay, I'm sorry. It looks red to me. Okay, so it's an orange ball, and it's being grabbed by four. It's being grabbed by two histidines up on top, and two cysteines on the bottom. And they grab that little ball, and they manage to fold the protein. So there's a knuckle here. You see it right above the N? That would be the knuckle. And that's what is going to have some magnetic attraction to a piece of DNA. And they've been able to take these zinc fingers and engineer them, and they're creating just absolutely perfect little motifs that will grab onto a specific piece of DNA. And Cell mentions that they're it's a big engineering problem. You have to have one hell of a lab to do it, and you create these little grabbers that can find a piece of DNA. Like if you take each of these little knuckles will identify 3 nucleotides. So the thing right above the N is called the knuckle, and the whole thing is like the finger. Well.
Oh, okay.
Yeah. Okay. So you bend your knuckle down and you grab 3 particular nucleotides. So a nucleotide sequence, CCA, something like that, right? And then you hook it up to another one right next to it. You duplicate that segment, and now you have something that'll grab a GGA. Well, now you can grab CCA, GGA. So they're very interested in this pharmaceutical industry because they can develop very specific gene finding mechanisms and hopefully at some point do something, cut the gene out, enhance it or whatever. So anyway, but in the cell, like the cell that Cell famously talks about, I think is the one he talked about here was the TF3A. That one, even though it has a number of fingers, only 3 of them matter. So it's searching for a sequence of 9 nucleotides that are present in the gene for a piece of the ribosome, and then it upregulates it. Now, that doesn't mean that it couldn't have been transcribed without that upregulator. It just means that it's going to be upregulated and it's going to be transcribed more, in greater numbers, right? So it's an enhancer, that sort of thing. So that's one of the things. But only 3 of those fingers mattered. Why are there 17 fingers when only 3 matter? Well, that's the way evolution works. It's messy. Yep. So anyway, and there's
parts.
Yeah, and there's other parts here that show you different motifs and different ways to create these things. So zinc fingers are kind of prevalent. So you got zinc binding loops, you got zinc 2 cysteine 6, you got a zinc ribbon, a whole bunch of stuff. Gag knuckle. I don't know what that one does, but each of these are worth looking into. So
these are just like protein shapes, basically.
Yeah, they're protein shapes. And the zinc, the zinc really stabilizes the shape. So it's a pretty stable protein because the zinc grabs, 4 different amino acids and clusters them together. And it has to do with the hybridization of the zinc ion. So, but there is just, there's just a lot of wonderful biochemistry involved here that we can look at forever and ever. And I have a lot of papers that I've clipped. I'll show you a classic zinc finger here. I'll stop screen. This is sort of a cartoon diagram. Where are we here? And I'm gonna pull a cell on you and show you my PowerPoint, which I use PowerPoint to take notes when I'm studying something. And so I have one on transcription or translation in general, and I just put a lot of stuff. But the green is the zinc ion, and you could see on the bottom there's 2 histidines, and on top there are 2 cysteines, and they have a little sulfur. And and I wanted Immutable to tell me more about histidine chemistry, but those little points between the 2 nitrogens grab onto that zinc. So, and then I have what else do I have? So I've got some different motifs. Here's the C2H2 and stuff, and different ways to look at them. And I got some papers, and here's the molecular orbitals and a whole bunch of other shit. I don't know. Why does my mouse? I got to buy a decent mouse. Yeah, there's the molecular orbitals of the zinc ion, and I don't know. So I got a bunch of stuff here.
Seems like it.
Yep. Anyway, fun to study.
Yeah, I definitely need to look more into this.
And I had a hell of a refresher course in all of this morning and yesterday. So, and you inspired it. So thank you. And Immutable, you really had a great conversation with Cell. And I was very pleased with Cell and what he did. And yeah, we'll leave it there.
And I'm glad I could be helpful in that. So
yeah, you guys did a great job of giving us stuff to review here.
Yep.
So I think we can end the stream.
Sure.
Abruptly, as we are known to do. Huh?
Well, we're telling people that we're about to end the stream, so you could get a little more abrupt.
I could.
You could just like in the in Andrew's middle of his sentence, you could just hit the end broadcast button, and people would be going, "What the heck?" But now they know you're about to end it all.
God damn! I get. Oh, well, that was a foreshadowing.
Oh,
Subtle
sorry.
Foreshadowing. Yeah. Goodbye, crew. Click. We're out. Goodbye, cruel world. We're ending the goddamn thing.
And thanks for being here.
Thank you all for being here. We managed to keep 14 to 17 people watching, so I guess it wasn't too bad. But we'll see if they come back for the more technical review of this stuff when I know something. And hopefully, cool. Italian chef holds zinc and touches the spaghetti with his knuckles. That's the visual you want. Thank you, CRISPR.
I'm hungry now. I'm looking at my Uber Eats because I want some pasta.
I want some pasta. Oh, I've got corned beef in the crock pot up there. I'm gonna make. I gotta go get a cabbage, make boiled dinner with it. That's happening now. Okay, goodbye, crew world. We're leaving.